two concepts. One is antigenic shift and antigenic drift. So a shift, as the word, as we say, just shift a bit. So when a small mutations happens within the same virus, it is called antigenic shift. But when these mutations keep on accumulating, they can become a new strain. That's how happened in each and every pandemic, including in the corona. So the influenza basically, human is an accidental host or not always a primary host. Certain strains of influenza grows very well in birds, like in ducks, in pigeons, and in pigs. So in many countries where pork and duck meat is consumed, so there they get a chance for these viruses to resort. And this is suspected what happened in Wuhan during the corona pandemic also. So when these viruses get a mixing bowl in form of two different species, they evolve and they become better adapted to infect other species. So here are some of the changes based upon that definition. What happens in a drift is a minor change within the subtype. It can be a point mutation, a T to C, adenine to thymine, or G to C. So it occurs within A and B subtypes, and it has a potential to cause epidemics, but not pandemic. But in a shift, a major change, a new subtype has emerged. And what happens is the gene segments have exchanged. When scientists found that these gene segments have exchanged, they used this knowledge to make pseudoviruses and reassortment viruses for predicting what could be the next strain when the new pandemic comes. So that's why when I said we are better prepared, we do pseudoviruses with complete ethical clearances. In some countries, it is still debatable, especially what happened in China. So having said that, what makes these viruses evolve is still being understood. There can be a genetic bottleneck. The word bottleneck is used because, as you see, the bottleneck is very thin, and only if something has to pass through, it has to pass through that thin area. So the genetic bottleneck is from mutations. Not all mutations are fruitful for the viruses to evolve. And there's also a bottleneck from the infections in forms of the species. So human being one of the most important species, it's not always that all humans who get infected are diseased and they allow the replication to happen. So these two bottlenecks together, if they are continuously studied, we understand how the viruses evolve. So this is one of the best examples to understand the evolution of viruses. The next example which I'll be dealing with is HIV. So now I have spent some good time on making you understand how the pandemics of influenza created a havoc. But since the title says it's historical, so here, when the first pandemic happened, we absolutely had no idea how to combat. There were only antibiotics available, but antibiotics were not working. And there was a huge hue and cry among the countries why they are not working. So when I introduced the egg-based vaccines, so in 1940s to 1950s, the egg-based vaccine was very prominent. But egg has albumin, and albumin causes adverse drug reactions to humans. At that time, there was always a question of cost to benefit ratio. So apart from having adverse drug reactions, the egg-based vaccines were thoroughly made, and it was mandate in many countries to give them to children. But children were falling sick after a certain time, and they were having adverse drug reaction and anaphylactic shock. The scientists understood that albumin causes some severe reaction in humans, so they started shifting their base to using cell-based vaccines. So only after the second pandemic in 1950s, the cell-based vaccines started prominently being studied. So here you see a flow chart about how in the egg-based vaccines and in the cell-based vaccines, the viruses are grown, and now we have a technology. And just to tell you, this is not the egg which we get in the market to eat. This is an egg which is embryonated. And now the World Health Organization says whatever eggs which we use as a manufacturer to produce, these have to be certified and they should be specific pathogen free. Back then, when I was working on this, it was 10 years ago, one egg cost 280 rupees. So these are very thoroughly made, and the birds which are making these eggs are given complete reverse osmosis water without any bacteria in it and antibiotics. So manufacturing of vaccine becomes a very costly process, but also it has to go through a lot of channels where we make sure the end user, the human, is not suffering. So now there are guidelines which every country, as mandated by the World Health Organization or CDC, Center for Disease, have to be followed. Parallel to the egg-based vaccines, there is cell-based vaccines. One of the most earliest form of cell culture-based vaccine which has been used was using MDCK cell line. For most of you, you may not know, this is Derby canine kidney cell line. So it comes from canine, that is dog, the source. So these cell lines were adopted in the lab and they were transformed and they keep on growing. And in that cell lines, the virus is inoculated and grown. And then it is scaled up using fermenters. So currently, since many countries have found that the young children are getting annual influenza issues every year, they fall sick, and even we say that then it's a change of season. So what the World Health Organization and many countries, including India, is we share the surveillance report. My alumni mater, NIV, was one of the surveillance center recognized by WHO. So whatever the virus strains are being collected from clinical samples are sent to the surveillance centers, and they are sequenced, and they share this information with WHO. Based upon this information, in the northern hemisphere, that is above the equator, and southern hemisphere below the equator, the WHO releases that this is the particular circulating strain this year. So all the vaccine companies which are making seasonal flu vaccines, they use this information and they have a flow chart. So right in the month of Feb, right now where we are, WHO will release the strain information. And no matter what approach you can use classical egg-based or reverse genetics-based, where you reassort the virus, or in cell culture-based. By March, April, they start to grow this virus in such a way that by the next season, the next winter season, the vaccine is available in the market. It looks very simple and beautiful in the picture, but it goes through a lot of rigid, stringent conditions, and there cannot be any mutations happen. So in the virus, so these has to be tested. So it's a very full-on work, and this goes on 365 days. So this is one of the best example. As a student of virology and immunology, we are taught how to plan your end product, both in terms of research, in terms of business. So one of the major companies in the world which are making this flu vaccine are having a lot of profits, because in the Western countries, the flu vaccine is given seasonally to young children and to old people. So what would we do when we have to prepare for a next pandemic? So now I hope I have imbibed some of the knowledge, how the knowledge of immunology, virology, the classic microbiology is important. So if one way is the preparation of pseudovirus particles. So pseudovirus particles are nothing but it has a structure which doesn't allow it to replicate, but it has a genetic material inside. These can be prepared in advance and kept, and the seed strain can be verified, and then they'll be tested as a potential vaccine in animals, like in mice, in guinea pigs, in rabbits. And we can optimize the growth of the virus, and then we can prepare a seed and make it for the bulk vaccine manufacturation. So this is a review from Nature's paper, a very well-reputed journal, and it has advised, and it is actually appreciated by World Health Organization, this group of people who have given this, and they say that this should be the way we should be preparing for a next pandemic. One of the other viruses which I would like to take your attention is the HIV virus. The earliest discovery of HIV virus in humans was in 1980s. In India, it was 1988. The first patient was identified in Goa. So this is an example of how the virus replicates. Like right from the first part, when the virus is identified by the host cell receptor, it is engulfed inside, and once it is engulfed, the RNA of the virus, becomes naked. And you should remember that the viruses don't have their own mechanism to replicate. They always they steal the host mechanism to replicate. So inside the host, the mRNA is converted into protein, but it is not a simple step. While studying the replication of HIV, two scientists, one in France and one in US, they found that they have an enzyme called reverse transcriptase that transcribes the RNA into DNA, and then the DNA integrates to the host cell. Now, this window period of transcription from RNA to DNA and the integration, this is very crucial because most of the HIV drugs or vaccines focuses on this part. Because once the virus integrates into the host cell, there's no way for the host to identify whether this is our own genome or the virus genome. And this exactly becomes one of the reasons, if not all, why we cannot make HIV vaccine. The other reason is each time the virus replicates, it takes some part of the host surface proteins. And because of that, our antibodies, which are circulating, are not able to identify that the new virus, which contains some of the host proteins on the outer surface, is actually non-self. So this is still very difficult. And to best of my knowledge, there have been so many trials for HIV vaccine. So far, the only one closest which we can have is a HIV vaccine trial happening in Thailand and India, which is in phase 3, but it has only shown 50% efficiency. So we are still debatable whether this vaccine will be available for humans to use. Having talked a lot about vaccine, I will also dwell into how we classify the vaccine. So this is a screenshot from the Ministry of Health of USA. So this is, we have different types of vaccine. Let's start with subunit vaccine. So the subunit vaccine is part of the vaccine, as it's called subunit. The active ingredient of the genetic material or the surface protein specifically is used as the part which can give the immunity. Then there is nucleic acid vaccine like mRNA vaccine. The inactivated vaccine could be the polio vaccine, which is inactivated by heat or formalin. There is live vector vaccine using adenoviruses. And there are virus-like particles vaccine, which is very new. We are still understanding. And there is gene-deleted vaccine. If you remember, I talked about dengue. So now the current dengue vaccines, which most countries are making, are using a deletion of one of its surface protein, which makes it weakened. And this deletion is used to grow and then prepare vaccine. Just to pick your brains again, this is an example of the type of vaccines. There is live or weakened. One of the examples are the measles, mumps, rubella, which is given as a trivalent MMR vaccine, and it was first introduced in 1798, and amazingly it still holds good even now. We have kids, we are inoculating them with this MMR, and MMR antibodies, especially for mumps, they only stay for 10 years. So some people, they suggest that we should have a booster dose after 10 years. There could be a kill whole vaccine, which can be polio, Japanese encephalitis. Then there's toxoid, diphtheria, and tetanus, which is now given as a pentavalent along with measles, mumps, rubella. India is the largest manufacturer, and Serum Institute of India is one of the largest manufacturers, which are making this amazing work of making childhood vaccination. And it will be surprised to know that they make this vaccine for $1 per dose. Even though they are making profit, but they're selling it only for $1 per dose, and they sell it to African countries. And then there is a subunit vaccine like for pertussis, for meningococcus, and anthrax, which is a very important post 9/11 because of the bioterrorism. So anthrax vaccine is also a subunit vaccine. Now coming down, there is the mRNA vaccine like the SARS corona vaccine. So this mRNA vaccine technology has revolutionized how we approach to beat the time frame during the pandemicity. So now most of the research is happening on mRNA vaccines, especially after